Hi, my name is Sidney Martin. I'm a rising junior that attends the University of Alabama in Huntsville. Today, I'm going to talk to you about imbalanced muscle lipid metabolism in LU1WR1 rats, which results in increased mass and reduced insulin sensitivity. So first, I'm going to talk to you about type 1 diabetes. So in a type 1 diabetic person, immune cells attack beta cells that produce insulin, and that results in increased glucose blood levels. So the Love Rutledge lab found that in FAT10, in the upregulation of FAT10, that leads to obesity, kidney disease, and type 1 diabetes susceptibility. The lab also found a rat model that has the same characteristics. So with that in mind, we wanted to see how FAT10 affects biological pathways. So first we had our control rat and we had our experimental rat, LU1WR1. And with both of those rats, we had a high-fat diet and a control diet. And as the study progressed, we saw that the experimental rat had more mass than the control rat. And we wanted to see why. So we extracted muscle tissue, isolated the RNA, and converted that into cDNA so we can perform qPCR and see why the experimental rat had more mass than the control rat. So first we tested lipolytic genes. And lipolytic basically means the ability to break down lipid. As you can see in both the control and high-fat diet, there was a downregulation in lipolytic genes. On the contrary, we tested lipogenic genes, which basically means the ability to produce lipid. As you can see, the lipogenic genes were upregulated, which means that in the experimental rat, there was more lipid being produced than being broken down, which leads to increased mass and reduced insulin sensitivity. We also tested GAPDH, which is involved in both glucose pathways. However, because we only tested GAPDH, the results were inconclusive. So in future studies, we want to see how FAT10 affects the glucose pathways within the muscle tissue. Thank you so much.